Welcome to Biology Minds. Today we're going to talk about biochemistry, the chemistry that involves biology, living things. So just as a basic reminder, some of you may have learned this previously, atoms are what make up everything. So just like maybe you've heard that cells make up all living things, well, atoms make up all living things and everything else as well. It consists of a proton, a neutron, and electrons. Protons are going to be positively charged, okay? Neutrons are going to be negatively charged, designated as a zero, meaning there's not positive, it's not negative. And then electrons are negatively charged. We have the protons and the neutrons. These are both in the nucleus. And then we have the electrons in the electron cloud. Now, when we talk about chemicals and chemistry and atoms, when we have more than one atom, okay, put together, we call them molecules. If we have a bunch of atoms put together that are all the same atom, we call them an element. So if you hear someone say a gold element, okay, that's a lot of gold atoms. Everything in that piece of gold is a gold atom. Compounds made of two or more different atoms. Now, chemical bonds, there's different types of chemical bonds. All right, it's a force of attraction between different atoms that hold them together, which obviously forms a compound. Okay, we have different atoms, they're coming together, they form compounds, and they're held together by chemical bonds. When we talk about chemical bonds, you're going to hear a lot about non-polar covalent bonds and polar covalent bonds. Now, this word covalent means that they're sharing. Okay, they're sharing electrons. All right, electrons. Okay, we have our atom. Okay, and we'll have our proton, and we'll have our neutrons. Okay, and we'll also have our electrons on the outside. All right, in our electron cloud. Okay, and these electrons, they can either be shared, okay, or they can be given and received, like in ionic bonds. Now, the difference between non-polar covalent bonds and polar covalent bonds is that in non-polar covalent bonds, there's going to be an equal sharing, okay? The atoms are sharing it equally. With polar covalent bonds, it's unequal sharing. One atom is taking control of it more than the next atom. So this is just a representation. You have your non-polar covalent bond. Okay, you see that there's an even sharing and equal sharing between the two atoms. Down here we have a polar covalent bond where you see that the chloride atom is taking more control of these electrons than the hydrogen is. All right, and then you have ionic bonds where this sodium is going to give an electron, okay, to the chlorine or the chloride atom. Hydrogen bonds are going to be very important later on in the year when we talk about DNA and we talk about nucleotides, okay? But just understand for now that they are weak bonds, okay? It's going to come into play later. It's important in water, DNA, and proteins. When we talk about pH, that's ultimately what I tell students, it's the amount of hydrogen ions within a solute, okay, or within a solution. Now, when we think about acids, we think about maybe taste, okay, taste sour, so you talk about, oh, a lemon is very acidic. Maybe when you talk about bases, you talk about certain soaps or cleaners, all right, they typically are good cleaners. And when you talk about acids, maybe you talk about hydrochloric acid, sulfuric acid. You hear about these. These are very strong acids. You don't want to play around with these. But we know that anything from 0 to 6, right? Typically, you know, we say 1 to 6. It's really 0 all the way up to 6.9999 and so on and so forth. All right. 7 is going to be neutral. And then we have pH of 7.0 0, 0, 0, 0, 0, all the way until we finally hit some 1, some imaginary 1, all the way up to 14. That is going to be basic. All right, we have acidic, we have our neutral at 7, and we have basic. Now, a human body is typically 7.35 to 7.45. So it's relatively neutral, but it's more towards the basic side. Just something to keep in mind for later on in the year. What you will have to know is the difference between inorganic and organic molecules. Now, what I tell students is always remember, if it has carbon and hydrogen, then it is organic. Okay, so there's plenty of things that have one or the other, but if they don't have both together, then it is inorganic. Okay, oftentimes you're also going to see in these organic molecules that have carbon and hydrogen, you'll also see oxygen, but it doesn't have to have oxygen to be organic, but it does need carbon and it needs to have that hydrogen. So something like H2O, students always say, but it has the hydrogen. No, it needs the carbon as well. So something like C6H12O6, all right, that's glucose. That's glucose. That is a sugar. And that is an organic molecule, organic compound, because it has our carbon, it has our hydrogen, and look at that, it even has our oxygen. This is the periodic table of elements. Okay, you see that carbon sits right here. Okay, and when you go on to learn in chemistry, you're going to see that the reason it sits here is because of its electron cloud and the number of protons and electrons it has, and that plays a big part in how it fits in with bonding with other molecules. But we won't go into that today. This just shows you the electron cloud. If it only has one electron, you're going to see it's going to just circle around that nucleus. If it has two, you're going to have two go around the nucleus. But since we do call it a cloud because there are different layers. So once you get more electrons, okay, you're going to fill up 2, okay, then eventually 8, and it goes on and on from there. So it's not just one ring around it if you have more than 2 electrons. There's going to be 2 rings, eventually you'll have 3 rings as you see down here, and so on and so forth. Now when we talk about organic molecules, all right, there are 4 types. Okay, this is number 1 that we're going to talk about. Carbohydrates, okay, the building blocks are monosaccharides. Now mono means one, okay, one sugar. A long chain of these monosaccharides is going to be called a polysaccharide. All right. Main function, energy. All right. So if you ever hear someone say, "Oh, I'm carb loading," or "I'm eating a lot of carbs," okay, that's because they want a lot of energy for that race they're going to run or whatever sport they're about to play. So they need energy, so they're going to take in a lot of carbs. Carbs are our sugars. Right. There's a lot of sugar when we talk about bread, pasta. Dairy has a lot of sugar. These are all carbs. Examples, glucose. Okay, glucose is the sugar that our body runs on. Sucrose, this is table sugar. Okay, our body breaks that up into glucose and galactose, which are sugars that our body can use. This is a basic example of a carbohydrate. We have glucose here. Okay, this is a 6-carbon sugar, makes this hexagon. Okay, sometimes it might make a pentagon, but it has 6 carbons, 12 hydrogens, and 6 oxygen. Now lipids, okay, typically we're going to call these our fats. These are anything that is considered a fat, an oil, or even waxes are all lipids. So when you hear that word lipid, okay, you should think fats. Now, structurally, there's not necessarily monomers or building blocks like we're going to talk about for proteins, like we said for carbohydrates, but they are made up of 3 fatty acids, and these 3 fatty acids are connected by a glycerol. Several functions to lipids, right? They cushion the body. So if anyone ever says, you know, it's good to have a little bit of fat. Well, you want some cushion for your body. You want to have something that's going to protect your organs. So it's good to have a little body fat. Also, functions in your cell membrane, very important. We'll get into that later on in the year. This is the glycerol backbone right here. All right, maybe I've heard someone say glycerol backbone. Part of my handwriting. And then these are my 3 fatty acids. All right. So there's different types of lipids that you've heard. I'm sure you've heard about trans fats, saturated fats. Okay, unsaturated fats, these are liquid at room temperature. They're better for you. Right? So if you're going to take in fat, you want those unsaturated fats. They're usually from plants, okay, nuts and seeds. They contain double bonds that create gaps. They have less hydrogen atoms. Okay, it's a lighter molecule is what I tell my students. It's a lighter molecule. Then you have your saturated fats. These are solid at room temperature. Not good. Okay, usually in meats. Okay, it's still okay to take in saturated fats, not the end of the world, but it's better to take in unsaturated fats. They do not contain the double bonds and they're heavier molecules. Okay, your body can still use them, but we don't want to take in too much. Okay, increased cholesterol, increased fat intake, no good for your body. We also have polyunsaturated fats, unsaturated fats with more than one double bond, but these are good for you. Okay, it's good for you. I know you're thinking, oh, more than one double bond, that sounds like a bad thing. Too much bonding? No, that's a good thing. Trans fats, these are the ones that maybe you've heard about in the past. All right, KFC, McDonald's, they stopped using trans fats because a lot of information was published about how bad they are for you. They are man-made unsaturated fats. So we said unsaturated fats, good for you. But these are the man-made ones. Our body's not used to using these, and they're not good for you. So you want to avoid them. And for the most part, major chains, major restaurants, they're not using them anymore because customers don't want them. We now know that they're no good for you. Proteins, these building blocks are monomers. Okay, you're going to hear this word monomer, and you might say, I don't know what that is. That just means building block. Okay, sometimes students they say, I don't know what building block is. What do you mean a building block? All right, well, if I was going to build a house that was a protein, okay, I'd use all these different bricks, and my bricks would be my amino acids. So that's all we mean by building blocks. It's not nothing deeper than that. But monomer is that word that we're going to use. You're not really going to hear bricks to the house or building blocks too often. You're going to hear what's the monomer for proteins, all right, amino acids. What's the monomer for carbohydrates? Monosaccharides. When we form these long chains of amino acids, we call them our proteins. All right, simple enough. Function, growth, and repair, very important in muscles. They also, some examples are enzymes, and as we go through the year, you're going to learn about the importance of enzymes and the role that they play in the body. And also other examples, hemoglobin is a protein as well. Proteins have a unique structure to them. Okay, they're all going to have this amino group. They're going to have hydrogen, just a hydrogen atom, a carboxyl group, all right, and then they're going to have this chain that changes. So this we call it the R chain, but it's different on each different type of amino acid. Okay, so these will always be the same. These three right here, our amino group, our hydrogen atom, our carboxyl group, but the R chain will change. So that R right there, that's not an R atom. All right, that's just a representation of a chain of other molecules that can be there. Proteins have 4 levels of structure. Okay, they're going to have the primary, all right, just a chain of amino acids. Okay, you're going to have your secondary. Okay, that's either going to be an alpha helix or a beta sheet. Then you're going to have your tertiary, where it's going to be those alpha helixes together or the beta sheets put together. And then these tertiary structures put together are going to form our quaternary structure. Here you go. You have your amino acid chain. That's our primary. Okay, then you have them form this secondary structure where it's either going to be a pleated sheet or a beta pleated sheet or a alpha helix, depending on the protein. And then when you put them together, you are going to end up with this tertiary structure. Okay, this tertiary structure has both pleated sheets and alpha helixes. And then when you put all these different tertiary structures, you're going to end up with your quaternary protein structure. Our fourth and final organic molecule, okay, there's only 4 kinds. We talk about carbohydrates, lipids, proteins, and now nucleic acids. Now, nucleic acids are important because ultimately they're the instructions for building our structures in our body. All right, they dictate how we are made up, what we are. Okay, their building blocks are nucleotides, and you'll hear more about those later in the year when you talk about DNA. And their polymer or their chain is nucleic acids, right? So nucleotides make nucleic acids, and they function as the instructions. I always tell students they're the instructions, okay? Depending on which organism you are, some have DNA, some have RNA, okay? We have DNA, we also use RNA, and you'll hear more about that in the future. Okay, nucleic acids, these are our nucleotides right here. Okay, so it's gonna be made up of a sugar, phosphate group, okay, and then you're also gonna have nucleotides. That's a nucleotide, this is a nucleotide, this is a nucleotide, and this is a nucleotide. So enzymes are organic catalysts. This catalyst just means that it helps speed up chemical reactions. Okay, usually enzymes end in ASE. So if you hear something like polymerase, ends in ASE. That is an enzyme. Okay, maltose. In order to break up maltose, we're going to use maltase. All right, ends in ASE. So the shape and its function are affected by 2 major factors: temperature and pH. Okay, if the temperature is too high or the temperature is too low, the pH is too high, the pH is too low, it's going to denature an enzyme. When it denatures an enzyme, it will no longer be able to function the way it properly should. Enzymes are specific to one substrate, so don't just think that, oh, and there's one type of enzyme in your body and it takes care of all the things we're going to talk about this year that enzymes do. No, there's one enzyme for one substrate. Maltase only works for maltose. It doesn't work for lipase or protease or helicase. It only works for maltose. Maltase, maltose. They must fit with their substrate. We call this the lock and key model. So they have to fit together. Their one enzyme is specific to the one substrate. Okay, we talk about synthesis reaction. Okay, dehydration synthesis. You're going to remove a hydrogen from one molecule, a OH from another molecule, and that's going to put them together. All right, so now our ABC and our DE come together to be ABCDE, and then we're left with a water molecule because we had our H and we had our OH. All right, now this ABCDE, this is just a representation. There's no molecule named ABCDE. All right, and then down here we see a decomposition reaction or a digestion reaction. We call that hydrolysis. And that's where we're breaking up molecules by adding an H to part of it and adding an OH to the other part. So we added H to our ABC, an OH to our DE, put water in, and we end up with these 2 different molecules.